for our third year of this event. We're always impressed and inspired by the diverse group of attendees and thoughtful engagement. PGTME is a project of the Division of Medical Ethics, housed in NYU Grossman School of Medicine's Department of Population Health. You can learn more about PGTME on our webpage. Kara is going to drop a link on the chat so everyone can see it. And you can sign up for our quarterly newsletter to stay abreast of our work. As a reminder, if you are unable to attend any of our talks, we will be posting captioned recordings once the week is concluded, and we'll share the links with all the attendees. We want to highlight tomorrow's panel, Ask a Family Member, where you're able to hear directly from a gene therapy recipient and family members of recipients about their personal experiences. Today's topic is Ask an Industry Member or Regulator. I'm excited to hear our fantastic speakers. Following brief introductions by our panelists, I'll moderate a discussion and we'll have time for your questions. We encourage you to submit questions through the Q&A feature throughout the panel. With that, I now ask that our panelists introduce themselves and their work. Let's start with you, Dr. Bryan.
Good afternoon. I'm Wilson Bryan. I'm Director of the Office of Tissues and Advanced Therapies, or OTAT, at the FDA. I trained as a neurologist specializing in neuromuscular disorders, and I've been at the FDA for most of the last 20 years.
Fantastic. Dr. James?
Hi, everyone. My name is Emma James. I'm Head of Medical and Patient Affairs at Encodia Therapeutics. It's a biotech company developing gene therapies for pediatric CNS disorders. I joined the PGTME working group almost about 2 years ago. I'm from Wales originally, which explains why you can't understand what I'm saying, and also that I'm not very good at soccer. I did my PhD in medical genetics from the University of Oxford and have spent the past almost 20 years in drug development for rare pediatric genetic disorders. For the past decade, I've specifically focused on clinical development of gene therapies with a specific interest in medical and patient education to incorporate the insights into the clinical trial design, ensure appropriate education, and make sure that the communities are able to make informed decisions, and also to improve data dissemination communication. I also wanted to add that my opinions expressed on the call today are my own. They're not representative of Encoded necessarily.
Thank you. And Dr. LaRosa?
Yes, hi, good afternoon. Greg LaRosa. I'm a Vice President of Head of Scientific Research in the Rare Disease Research Unit at Pfizer, located up in Cambridge, Massachusetts. I've been there about 10 years, a little over 10 years actually, and prior to that was in a number of small biotech companies in the Cambridge, Massachusetts area, focused mostly on inflammation and immunology, small molecule and antibody development. But being at Pfizer in the rare disease group, we've embraced for about 6-7 years now gene therapy, and have several clinical trials, have 3 phase 3 trials going right now. And I'm happy to participate in this and hopefully answer some questions.
Fantastic. We're going to start with a question for you, and I want to encourage all the panelists to chime in anytime they want to add a comment. So we'll make this very interactive. So the first question, Dr. LaRosa, is how does industry select therapeutic areas to pursue for early gene therapies research?
Well, I think it's probably depends. Different companies probably do it somewhat differently, but typically we're focused in basically 4 main therapeutic areas within rare disease: rare non-malignant hematology, rare neuro and neuromuscular, rare cardio, and a little bit of rare metabolic. So first of all, that's our strategy. So we're not going to consider going outside of those 4 areas. And then within those areas, though, there's, usually we're take into account the the level of unmet need, which in rare disease is often quite a lot, but not always. But so, level of unmet need, and also the level of scientific understanding of the disease and the disease process. Often with rare diseases, we know what the genetic defect is, but that doesn't always explain how to go about solving or treating the disease. But with gene therapy, often it's a gene replacement or a gene knockdown sort of approach. But it really depends on how well we understand the biology and the clinical path too, which often is not that well known in rare disease space. And then there is an aspect of the commercial opportunity. I mean, it has to, as a public entity, there is a question about, does this make sense from a commercial and return on investment point of view? So, ultra rare diseases are very difficult to fund internally versus many of the other projects that are going on across the company. So I think those are sort of the main things, but I'd be interested to hear, Emma, what they do at Encoded, but I think that's generally what we try to do. And I think it's probably similar across the industry.
Thank you. And Dr. Bryan has a comment?
Oh, yeah, Dr. LaRosa, this is of great interest, I think, to all of us, because gene therapy now, we think, has the potential to address so many of these extremely rare diseases that are caused by single gene defects, but the populations are just so small that we worry that it's not, at the moment, commercially viable. And I'm wondering, is there you probably had a lot of internal discussions in your company, is there a number that seems like too few patients at the moment? And do you see that changing in the future? Is the technology going to get us to a point where we really can attack those diseases that affect 4 or 5 people?
Right. Yeah, 4 or 5 people is, probably too little for, some larger pharma companies, I would imagine. One thing, that we're part of, Pfizer's a part of, along with some other companies, is the Foundation for NIH collaboration, a consortium among a number of companies called the Bespoke Gene Therapy Consortium, BGTC. And the aim there really is to try to simplify the pathway, the development, the research and development pathway, as well as the regulatory pathway, to perhaps bring the cost of delivery down, but also specifically to then pressure test what the consortium comes up with and actually run some clinical trials in ultra rare diseases, of which have no commercial, currently no commercial interest. And I think it's very important that, industry embraces that. And there is a way such as that, to find treatments for some of these, very rare diseases, outside of the normal, pharma construct.
Sorry.
Go ahead.
I was just going to say, I do think there's a lot of work underway to improve manufacturing, bring down the cost of goods, improve potency, which I think will of course enable smaller populations to be treated while still being commercially viable. It may still be that the ultra rare populations need alternative mechanisms than commercial entities. So, for example, maybe the foundation of N lorem, which is looking at N of 1 studies. Maybe there are other mechanisms that could eventually be put in place to help those populations, which of course do still have a significant unmet need. I think just one other thing I would add to what you'd said, Greg, originally was, I think it's not only commercial viability, but one of the things that we assess is that clinical trial viability. And I've certainly worked in spaces where we have initiated programs and then just not actually been able to design and enroll clinical trials. And so, understanding that disease epidemiology and the patient population, and whether we actually really can study something in that way is a really important first step in considering whether to start a new program.
And Dr. James, following up on that, what are the key factors sponsors consider when designing those first in-human studies?
Well, I could spend the rest of the panel talking about that. I do think, as we assess that feasibility, we spend a lot of time engaging with experts in the medical and patient and the regulatory communities to really understand how we go about designing the robust, meaningful trials with as little burden as possible. I guess I sort of think about four main buckets. There's the feasibility, which we mentioned. There's disease presentation and progression, there's dose selection, and then what is the precedent, the outcomes, the endpoints. So in terms of feasibility, we start by identifying experts at institutions with infrastructure, expertise, experience with clinical trials, and ideally gene therapy too. We try to marry that with who the patient community knows and trusts, but it doesn't always align with that gene therapy expertise, at least currently. We've mentioned the unmet medical need. We know that there's a lot of urgency here, so we're trying to initiate trials as quickly as possible. So we do look at institutions and countries that have faster startup timelines. And then, as I said, the disease epidemiology, so where the patient's located, how they're diagnosed, what are the disease characteristics, what approved treatments are available, what's the burden from both the medical and the patient community. In terms of understanding disease presentation, we want to understand, does the disease pathophysiology actually lend itself to the potential benefit of gene therapy? Do we know that the gene mutations cause the pathology? Is there a lot of secondary injury as a component of the disease that may not be able to be targeted? How do the symptoms evolve over time? Is there an unmet need? How do we capture change in that disease? And is it going to be possible over the time course of the clinical trial? And I do think that's something we really spend a lot of time thinking about in these diseases with very, very high mortality or significant severity in children. Many CNS or neuromuscular disorders have neurodevelopmental or degenerative features, and so you need that earlier treatment for optimal outcomes. But that really turns the traditional drug development paradigm on its head, where you have to start in children and to get that proof of concept, and then move into adults versus the other way around. And that requires a lot of conversations and discussions and thought about the appropriateness of that. Obviously, dose selection, it's not my area of expertise, but when you're thinking about designing a clinical trial, for gene therapy, it's a single administration. You can only give it once, at least currently. And so we spend a lot of time looking at the preclinical data and trying to determine what is that first dose that is going to be safe, but also going to provide some therapeutic benefit to the patients. And then in terms of the outcomes and the endpoints, we're looking at again, understanding the disease so that we can determine appropriate outcomes and endpoints that are meaningful to the patients, that reflect the nature of the disease, that they're going to be sensitive to change over the time course of that clinical trial. We need them to be able to be reliably measured across different trial sites and to be able to give a rapid indication of the effect on the relevant aspects of the disease before we go about dosing too many more patients again, because it's important to remember you can't take this away. And so we're really trying to learn from each patient as they are treated to inform the dosing of subsequent patients. And then I think the last thing that, and then I'll let other people chime in, is really, we spend a lot of time trying to define what that study population is. Often in these heterogeneous diseases, we have to narrow in on a small subset so that we can obtain results where we can understand what they mean and attribute whether the data is due to the results of the treatment or not. But we're also trying to obtain information that means that we can generalize the results to the wider population. I do know that this is very frustrating to patients and families who feel like they're being left out. I hope everyone understands that we're trying to do this in a thoughtful, safe way, but we are trying to develop therapies that will be impactful to the whole community. And so I guess fundamentally, the gene therapy clinical trials are just different, and they're very, very complex. And I think every sponsor coming into this space should be thinking about that education and engagement and ongoing communication with all of the different stakeholders to make sure that everybody is getting information as it happens and able to make those informed decisions.
Fantastic. And Dr. Bryan, you have your hand raised.
Yeah, it seems to me that you, Dr. James, and Dr. LaRosa have a very difficult job. There's a lot of excitement about gene therapy because the science has moved forward so much that we have the potential to have products that have a huge effect. But that's not always what we see. And that gets to the issue that you raised, Dr. James, about clinical trial viability. And if you have a huge effect, then the design of the clinical trial and the number of subjects starts to become pretty easy. But patients are interested in, and we're all interested in, things that have modest effects. Those matter too, and it would be very important for patients. But in these very rare diseases, how to have clinical trial viability in that setting with modest effects and rare diseases, and of course you don't know until you start in humans exactly what sort of effect you're going to see. So it seems to me again that you have a very challenging situation on your hands.
And Dr. Bryan, within the same area, how do you think the regulatory pathway differs between gene therapy and more traditional therapeutic approaches?
So from a regulatory standpoint, when we talk about different modalities of medical products, we talk about small molecules, we talk about therapeutic proteins, cell therapies, gene therapies, sometimes, and of course, devices. Many devices out there that help patients. From a regulatory standpoint, at least in the United States Food and Drug Administration, the regulatory pathway for gene therapies is the same as for therapeutic proteins and small molecules. It's different than devices. Devices have a very different regulations, but it's the same for gene therapies as for most other drugs that we think about.
And are there specific challenges that come to mind in relationship to therapy?
There are so many challenges, and some of them we've touched on already. That part of the excitement about gene therapy is there's so many bad diseases out there that are genetically defined, and these are diseases where parents would take their kid in to see the doctor, and 20 years ago the doctor would say, I don't know what this is. And I can't do anything about it. And now we have genetic sequencing, and now the doctor can tell you what it is, but we can't do anything about it. And I think we all think that we're going to fix those diseases. We're going to fix them. I don't know how long it's going to take, but the technology is coming along, and that's very exciting. But it takes time, and among the challenges that we see among drug developers have particularly been manufacturing issues, consistent manufacturing, issues around potency and comparability of products as they change during stages of development. The CMC issues have really been, in many ways, a greater challenge than the clinical trial issues for many of the products that we've seen.
Mm-hmm. Appreciate that. Dr. LaRosa, I was wondering if you could comment. Dr. James mentioned the issue of dosing, and I was wondering if you could also comment on how the human dose is calculated from animal doses, and how do we ensure we balance safety with reaching a therapeutic dose?
Yeah, that's a very important but a very difficult question in a lot of ways. It's I mean, you obviously do animal studies to demonstrate efficacy. That's also often done in small animals, such as rodents. And one of the problems we've seen in the field is that a lot of times the viral capsid that's being used may work in mice, but doesn't go to the same tissues in primates or in other, higher animals. So that makes some things sometimes difficult. There's been a lot of work done in the field on identifying AAV capsids anyway that can work more consistently across species and can target more readily specific organs of interest other than just simply going to the liver, which is where things started out. But it's a very complex process of doing PK/PD modeling, pharmacokinetic and pharmacodynamic modeling from the disease model work that's done, and then going into the biodistribution and safety studies in non-human primates typically is what's done. And again, a lot of computer modeling basically, and scaling up to what likely the dose or the distribution and pharmacodynamic would be in humans. But with gene therapy, it's also incredibly important because, as I think Emma mentioned, you can't redose right now. So you go in with a dose into a patient, you really want to be in that space as best you can, that you're going to see you expect to see some benefit, but you're not going to, be in a range that could cause some, serious adverse effects. So it is a tricky thing, and I don't think we entirely have a good handle on how to do that. But nevertheless, the trials have gone forward, and I think, we've seen definitely positive results in some. We've seen some more difficult results in others, and it's still a bit, I think, this field is still early in its development. It's not, not like a small molecule drug, that, has been done for, many, many, many decades. So we're still really learning how to better do this, but it is a very important question. It's just not that straightforward at this point.
A lot of trial and error, yeah. Dr. Brian, Dr. James mentioned the challenges of identifying endpoints that can be sensitive enough to pick up an effect. Are there, from a regulatory perspective, any individual considerations in endpoint selection? Are there specific considerations because it's gene therapy?
And I say that the rundown that Dr. James gave of the characteristics of endpoints, I agree with. Those are all very important. There's actually nothing special about gene therapy. The clinical trials for gene therapy, in a lot of ways, now, there are some differences, particularly around things like you can only dose once. But in many ways, clinical trials for gene therapies are very similar to trials for small molecules. So we'll use the same endpoints. And as Dr. James said, what you really want to do is you want to characterize the disease. You want to find something that's clinically meaningful that shows the activity of your product and that matters to patients. But you also want to look at as many aspects of the disease as you can.
Fantastic. Yeah.
I guess I would just
add, Dr. Jane.
Oh, sorry, go ahead. I was just going to add. I was just going to add to that. I think one of the things that certainly, we face working in a space where you've got children who are growing and developing is often the endpoints that you are looking at, they differ by developmental ability or differ by age, and so you're trying to design trials that where children need to move through different assessments. And often, especially in the CNS, we just don't really know what is the full capacity for recovery or for change. And so you end up with these trials where we have a lot of tools to assess specific outcomes because specific tools haven't necessarily been validated within a specific disorder, which is particularly the case for rare diseases. And it can put a lot of burden on the patients and the families who can feel very frustrated that they're being asked to do assessments that look like they're doing the same thing over and over again, and often their child can't necessarily do all of those assessments. And I think it's important to understand, part of what we're doing is trying to understand which assessments are actually useful for measuring different endpoints at different ages in different groups. But sometimes we just don't know. We do try to look at the viability of different tools so that we select ones that are as minimally burdensome to the child or the families as possible, especially as they're likely to have to do it many times. But it is something that we are very cognizant of, that often, in these very complex heterogeneous diseases, we're asking a lot of the patients and the families. Hopefully, as we learn more, we'll be able to, add to that growing body of evidence and reduce the burden on them. But in the absence of validated assessments for those different groups, and absent a good way to really move between assessments of with different floor and ceiling effects in different age ranges, it's very hard to design very streamlined trials, unless again you end up focusing on a very, very narrow age range, which then brings its own problems in terms of generalizability. So it's a difficult one.
Yeah, the challenges here, we talked earlier about if the drug has a huge effect, then all that heterogeneity, maybe we can work around that and we'll be okay. But that's not always true. And Dr. James, you mentioned some of these diseases that are heterogeneous and slowly progressive and long-term. With gene therapy, it could be that you're going to have a huge effect, but you can't see it for 4 or 5 or 6 years. And how do we do clinical trials in that setting? And it's very difficult. But I believe we'll figure it out. But it's just a huge challenge at the moment.
Yeah, I completely agree. I obviously working in the space of the genetic epilepsies, we're looking for endpoints that, for example, you might expect the seizure component to change within weeks or months. But obviously a lot of the value to the patient community is really kind of that cognitive, behavioral, developmental aspects. And we don't really know what the time frame for those things are. So we're looking for measures that are sensitive so that we can even detect that small incremental change over a couple of years, hopefully. But as you say, we just don't really know. And that brings into play then kind of the ethics of How do you continue to run trials that are enrolling patients when you haven't actually really seen that long-term benefit? So again, we're trying to find those endpoints that do show that proof of concept, I guess, that the mechanism is at least working. And of course, as you both know, there's a lot of work going on to try and identify clinically relevant biomarkers that also provide that kind of insight and guidance as to what the next step should be.
There's also a lot of work in the field to try to get around what Wilson mentioned about the a very large effect would make the trials more doable and easy, but often we see more modest effects. There's a lot of work going on in the field to, as I said earlier, to make second, third generation AAV capsids that are much more potent at getting specifically to the tissue that's where they're needed, and working on better promoters to drive the gene expression from the gene therapy. So hopefully, as we'll move forward, we'll be in that position to see a little better, maybe more often see a larger effect, which you might expect from something where you're directly replacing a gene that's defective. But again, some of these diseases are very slowly developing, and so that's always going to be a problem as well, though. But hopefully, we'll see some more better results in the future.
Good. I'm seeing some questions popping into the Q&A. I just want to encourage the participants to put your questions in the Q&A. We are going to get to those very shortly. And Dr. James, in the spirit of learning information about trials, if a family is interested in learning more about a specific gene therapy, can they contact the company developing the therapy directly? If so, how would they go about that, or who else should they reach out to?
Sure. Well, firstly, we always suggest that families reach out to their doctors first if they're interested in learning more about a particular therapy or treatment. Their doctors know their medical history and can hopefully provide more relevant general information on potential gene therapies and other treatments. Patient advocacy organizations are a great resource. Not all doctors are well versed in gene therapy or specific gene therapies being developed for a given disorder, but companies and sponsors work very closely with the patient advocacy organizations to provide new information that can be then accessible to the whole community. And we try to make sure that the patient advocacy organizations have the latest information in a way that is suitable for dissemination. If families have specific questions on clinical trials, they can contact the clinical trial doctors, also known as principal investigators. They're usually listed on clinicaltrials.gov, and clinicaltrials.gov usually has the latest public information on clinical trial design. And then to learn more about a specific gene therapy, they can certainly contact the patient advocacy department of a company. We are often limited in what information can be provided from companies. But typically, if you want to contact patient advocacy, the information is usually listed on the company's website. It's often listed on the clinicaltrials.gov posting for a specific trial. If you can't find the information, you could try the general information on the website, and they can put you in touch with the patient advocacy team. Not every company, especially early stage ones, don't always have a dedicated patient advocacy team, but if available, they're usually a really good resource. If you don't know who the patient advocacy organizations are, or you don't know what to look for on clinicaltrials.gov. I will say, if you have specific questions about eligibility for clinical trials, families should really speak with their healthcare provider or the trial PI. Sponsoring companies aren't involved with determining the suitability or selecting patients for clinical trials. And so we don't know who the clinical trials participants are. And all we're going to do is be able to refer you back to your physician or the trial doctors. So, we will do our best to help you, but ultimately for trial eligibility, it needs to be the trial sites that you speak with.
Dr. LaRosa.
Yeah, I just like to second that about the patient advocacy groups. You know, we with all of our projects work very closely with keeping them informed as best we can. There are some, you know, you can't necessarily say everything with early projects, but to keep them well informed, and they are. And that's the nice thing in rare disease is the patient advocacy groups are amazing, and they're very strong, and they really know the science, as do a lot of the patients. But they're a great source of getting, you know, an understanding about certainly what is gene therapy, and then is it applicable, you know, to my or my child's or condition. And it's they really are a great resource. It's one thing that's, I think, very positive in the rare disease space.
That's a question we had for you as well. How does industry work with the patient community around the development of gene therapy?
Well, you know, again, I can't speak for all industry, but I think in general, many companies anyway, really engage quite early with patient advocacy groups when we're, you know, thinking about planning clinical trials, and even sometimes earlier during the discovery phase. But definitely when we're thinking about planning clinical trials, we have patient liaison folks that are part of the rare disease research unit at Pfizer, and they pull in you know, members of the advocacy group and sometimes patients as well, and talk about this is how this is what we're thinking about with the clinical trial, and get you know, get their thoughts and ideas, and of course then there's interaction with the regulatory agencies as well that, you know, drive some of that design. But we try to include the patients and the advocacy groups as early as we can, just to get a good understanding of what their you know, what the patient situation is, first of all, what they're dealing with. And we often have patients come in and talk to us about their story, and it's always very motivating.
I would completely agree. We typically, you know, we'll almost approach it in the same way as our initial engagement with the medical community. So, you know, we'll speak to the patient advocacy groups. We may, through the patient advocacy groups, identify some families who we can actually speak with directly, you know, understand the unmet medical need, the burden, what their lives are like, what clinically meaningful or what meaningful endpoints would be to them out of a clinical trial or a potential gene therapy product. But also, you know, we'll share with a small group of them, you know, perhaps what our protocol is, or what the schedule of assessments is, or what the clinical trial educational materials are, to really get their feedback again on the feasibility of participating in a clinical trial from the family perspective, so we can try and see how we can improve that experience for them. I think, you know, the space is really changing, where, you know, I think the patients and the families are rightly now really being seen as really crucial partners to even early stage drug development efforts.
That's music to my ears. Dr. Brian, how about regulators? How do they interact with the patient community?
So in a variety of ways, it's very similar to what you heard from the industry. There are what are called patient-focused drug development meetings, where patient groups come together and they're open to anyone, where patients talk about their experiences with their disease and what's important to them. And that helps us when we think about the design of clinical trials, and when we think about what should or should not be in product labels as well. Sometimes when we have advisory committee meetings, we'll have patient representatives. Sometimes during drug development, when pharmaceutical companies come to talk to us, they'll bring a patient or patient representative in. We have recently started, I think in May of 2021, we started a Regen Med Ed webinar series to focus on issues that we think are important for patients, caregivers, and patient advocates. So there's a lot of interaction, particularly with the advocacy groups, less with individual patients. And actually that's one reason that I particularly like the patient-focused drug development meetings, is we hear a lot from individual patients and caregivers.
Fantastic. I'm going to start pulling some questions from the audience, and if we don't have enough, we still have a long list that we can talk about, but just to bring some of the audience into this. What specific strategies could be used to incentivize industry to develop drugs in the less financially advantageous disease groups? Has this been done before, and was it successful? I'm thinking of the Orphan Drug Act, but curious as to what might come to mind for you.
Yeah, I mean, I think things like the Orphan Drug Act have been incredibly important to incentivizing drug development in the rare disease space. In terms of the very small populations, I think it is done. I think there are companies who do move into spaces where they're not really going to make money, but it is something that they have some kind of proof of concept that their therapy could be advantageous in that population, and maybe it enables bridging to other populations later, or expansion to larger populations. I think there is a lot of work done where companies do some preliminary work and then spin out the clinical development into foundations or nonprofits, etc. So there are examples. I think generally, and Greg might be able to speak to this better than I, the larger companies are going to be those who are better able to take on the ultra rare disorders, just because if you have commercial income, then you're better positioned to be able to do that versus companies that are still sort of fundraising or just in the development space.
Yeah, I think that, as I mentioned earlier, being part of this bespoke gene therapy consortium, which is geared towards manufacturing, streamlining manufacturing, making it more commercially viable to then go into these ultra rare conditions. I think that's one way that the larger companies can participate in this. But I think also there's opportunities, I think, to do manufacturing for foundations that then and we've heard about some very small trials, N of 1 kind of studies that I think the manufacturing is often a sticking point for those sort of things. And I think the companies can also participate by providing some manufacturing to allow those trials, to enable those trials.
Dr. Bryan, you had a comment?
Yeah. When I look at this question, it reminds me of what we hear so often from patients or their parents of, I've got this rare bad disease. How do I get a drug company interested or willing to take on my disease? And Dr. LaRosa and Dr. James know this better than I do. Drug development is not easy. Most things that you think about, you got an idea, those ideas don't work out. Most products that go into development, they don't work, and it costs a lot of time and a lot of money. Every product that makes it to market, you look back at the science, and people have been working on that for 10 to 20 years. And so what can the individual patient or family do to try to make drug development less risky? And one of the answers that we always give, and I'm not sure if it's true, but I'll ask Dr. Rose and Dr. James to comment, is we recommend that you do good natural history studies. Beating a disease that you don't understand is very hard. You need to understand the disease, and that means understanding the natural history, understanding the pathophysiology. So getting that work done, I think, probably makes it more attractive to the pharmaceutical industry to spend the resources to step into the area. If that work hasn't been done already, then they're taking an even greater risk. But I'll see what Dr. James and LaRosa say.
Yeah, I agree with you completely. The more that can be done to show that you understand the disease, to understand, to characterize, again, its presentation and its progression, showing how it's diagnosed and where the patients are, and things like disease concept models can be really helpful to help us understand what potential endpoints or outcomes could be if we were thinking about that. But yeah, really, any work that can be done to understand the disease and the population, I think is a great starting point for us, because it enables us to then really come in with a good picture of the pathophysiology of the disease to determine how one might approach solving that problem from a scientific perspective.
And I think we've also seen over the past several years, patient advocacy groups taking on doing natural history studies for their disease. Going back to the question of the patient, how can I get industry interested in tackling my disease? And that's a big help, getting that natural history data as we've been talking about. And so a lot of the patient groups are doing that, and that's very incredibly helpful.
And that brings up a question from the audience for Dr. Bryan. If you could please speak to the ethics around placebo-controlled clinical trials in degenerative pediatric diseases, particularly as it informs the acceptability to the FDA of disease natural history studies as external comparator arms.
Okay, so each development program is different. Each indication is different, and the trial design that's appropriate for that product and indication varies. We approved a product called Zolgensma for spinal muscular atrophy, a very rare disease, and the entire development program. They did 2 clinical trials. The entire development program took only 36 subjects, and their evidence of effectiveness was to an external control, a natural history study that had been done several years earlier. So that's an example where you didn't need a concurrent control, but the reason you didn't need a concurrent control was because the effect size was so large. And from the natural history studies, we were able to identify some outcome measures that were inconsistent with the natural history. For diseases that are highly heterogeneous, and when the effect size is small, then you need clinical trials that are going to be able to tell us, is there something happening here or nothing happening here? And in some of those cases, I think placebo is going to be essential for development. So I'm a big fan of placebo-controlled trials when they're needed, but they're not always needed, and you have to look at the individual indication and development program to see what's necessary.
Great, thank you. Another question from the audience. Could you please say more about determining dose, toxicity, and safety efficacy in phase 1 trials, especially in the context of early access to therapies and efforts to expedite therapies?
So I guess I'll take a stab. I mean, the initial first-in-human studies are really focused on understanding the dose and the toxicity, and safety is what we are measuring and monitoring for very, very closely. Obviously, because again, as we've talked about, the early therapeutic benefit and the single administration means we do want to capture information on efficacy in those first in human studies. But those first trials are really to determine, is the product safe? In terms of early access, I think then it's really important to remember that that gene therapy can't be taken away. So it is a single administration. If it works, that's tremendous. It's great. If it doesn't, it's very difficult to manage those safety events. And it's important that that anyone receiving gene therapy is doing so at an experienced site who knows what to look for, what to expect, and how to manage those potential events. So it does make the risk-benefit assessment very, very different, and particularly so in terms of early access. So there's obviously a lot of hope and expectation around the gene therapy space, and we all work in this because it's something where we can really have a big impact, and the knowledge base is moving so fast. But I do think in terms of early access, it's something that until we have results from a controlled clinical trial where we have some information on what it is going to look like in humans, because as Greg said, we don't necessarily know what's going to happen as you move from mice to non-human primates to humans. I think we need to be very careful about sort of very early access outside of the constraints of a clinical trial.
Thank you. Any other comments? Okay. A question, I'm just pulling it up here. The screen moved, I'm sorry. From each of your perspectives, what are the most challenging aspects of gene therapy development, and can patient communities help with those challenges?
I guess I'll take a stab at that first. I think we've touched on many of the challenges. Mainly I think it's just really understanding, what the dose is and what the safety profile will be in humans, and whether we have potent enough medicine that with all it's a complicated medicine made up of a number of different components, and that combined, is it potent enough to be able to drive a good, fairly large response. And as we've also mentioned too, the other thing where patient groups can help, I think, is understanding the natural history of the disease. If that's not already known, that's as what we've all, we've been talking about, that's a big stumbling block in a lot of disease areas where we don't really know the clinical pathway.
I agree with everything Greg said. I would also add, I think, as I've mentioned, I think this is a space where education is absolutely critical. That's, education of the doctors, of the trial sites, of the patient community. And having that education and then ongoing communication is critical here. So I think, having the patient communities actually working with sponsors to disseminate information so that the community is aware of the latest knowledge that's coming out, because it's changing rapidly. Again, we don't necessarily have mechanisms to reach everybody. So finding ways to really reach the communities through the advocacy organizations would be very, very helpful. I think the other thing is, we've talked about designing clinical trials. The other thing that we really, we struggle with is that long follow-up that's going to be needed to see some of the effects monitor for durability. So we're looking at clinical trials that are going to be at least 5 years long, maybe lifelong. Like, we don't really know yet. And so I think having the community really understand that they are contributing to scientific advancement beyond their own benefit, and that even after that single administration that they have at the beginning of a study, that continued follow-up over years is really, really going to be critical to being able to advance understanding within a therapeutic area, and then also to be able to extrapolate beyond. And I think it's going to be incumbent upon sponsors to design trials or follow-up mechanisms that limit the impact to the families as we become more certain as to what those effects are. But it really is going to take a lot of commitment from the community too, for us to be able to actually collect the information that we need to be able to make this more broadly accessible to a wider community.
Thank you, Dr. Bryant.
I want to reiterate what was just said. As Dr. LaRosa mentioned earlier, we're early in the field of gene therapy. There's going to come a day when we're going to have hundreds, maybe thousands of gene therapies on the market for all these diseases. And those products will have been on the market for 10 or 20 years, and we'll know what we're doing. Right now, we don't know what we're doing. We don't know what sort of safety events are going to show up 5, 10, 15 years down the road or more. And we also don't know, as Dr. James alluded to, how long the activity of these products at work, how long are they going to work? If I use a product to treat spinal muscular atrophy, and I'm sure it's working, well, how long is it going to work? And do I need to have some strategy for retreatment in a field where retreatment is problematic due to immunogenicity? So from both efficacy and safety issues, we really need to follow patients until in the decades ahead when we know what we're doing.
We really needed several more hours for this panel because there are so many good questions here, and I'm sorry we won't get to all of them, but
I'm not hearing the moderator.
I think we might have lost Claudia. Let me. I guess. It's assuming that Claudia has lost her internet. I think she was saying we. Are you back, Claudia? Okay, so I will take a note of the questions, and Cara, I don't know if we're able to get back to people or provide some kind of feedback elsewhere to be able to answer some of those questions offline.
Yeah, I can just hop in real quick. Oh, there's Claudia.
Yes, I don't know what happened. I'm sorry.
That's okay. But I just wanted to thank Claudia, thank you for moderating, and thank you to all of our panelists. And to Emma's point, we will be collecting all of the questions because we know we didn't get to all of them. And PGTME, our working group, will put together sort of an FAQ sheet or document of all the questions, which we'll then send around to our mailing list. And if you're not already on our mailing list, I will drop my email in the chat. Okay, I'll hand it back to you for last words, Claudia, but thanks again, everyone.
Just saying thank you, and be sure to tune in for tomorrow's panel, Ask a Family Member. On behalf of PGTME, thank you so much to our panelists, and enjoy the rest of your day. Thank you.
Thank you. Bye-bye.
Thank you.
Bye.